Everyone for jumping on the line. I'm really excited to kick off this series of discussions between the data and monitoring, observing, modeling, and prediction group. I think these are really important discussions, and I look forward to the Q&A as well as the presentation. My name is David Allen. I'm a program manager at NOAA in the Arctic Research Program that sits within the Global Ocean Monitoring and Observing Program. I'm also a co-lead for the Interagency Arctic Research Policy Committee's group called the Monitoring, Observing, Modeling, and Prediction, or MOMP Foundational Area. I'm joining this call from the ancestral homeland of the Piscataway and Anacostan peoples. And I'd also like to recognize my co-lead for this group, Sally McFarland from DOE, who is also on the line with us today. If you recall, the IARPC 2022 to 2026 research plan established the MOMP to integrate and improve monitoring, observing, modeling, and prediction efforts to advance Arctic research across the entire Arctic enterprise. So literally to serve as the foundation for all of these different activities. One of the specific goals of the MOMP is related to data and is specifically to encourage and implement fair, findable, accessible, interoperable, and reusable, and CARE principles, collective benefit, authority to control, responsibility, and ethics, data management principles in the Arctic. Under 1.1, we were intended to identify verified points of contact, agency champions, data practitioners, Arctic residents, indigenous organizations, and their areas of expertise and interest for working with the data team on exploring and implementing fair and care in Arctic data management. As part of this developing the points of contact and identify track representation across many axes of diversity, demographic, disciplines, and sectors, IARPC experience, career stage, and others to ensure a diverse and representative group of contributors. The data team will check in with these groups regularly to ensure the points of contact are up to date. So that's a little bit about MOMP and why we have this particular point of entry on data. Now I'd like to turn it back to Cindy, who will introduce the data team and our goals for the meeting, as well as our first speaker. Cindy, take it away.
David, yeah, hi everyone. My name is Cynthia Garcia, or Cindy. I'm a program manager with the Arctic Research Program, which sits within GOMO at NOAA. So I'm joining this call from the ancestral home of the Anacostan and Piscataway peoples here in Maryland and Washington, D.C. And yeah, I'm thrilled to introduce the data management team within the IARPC Arctic Research Plan framework. So the data co-leads include Lauren Decker, who's here in the call, Jordan Kazemi from USGS, also in the call, as well as Vanessa Raymond. So we are the data team, the co-leads for the data management foundational activity collaboration team. So the team and our members work on advancing the objectives and specific actions towards a more fair, David already described what fair means, as well as CARE data principle, as well as to provide the space to talk and share resources about how we can advance those principles. So for our meeting today, we will be focusing on the management of physical samples in Arctic research. We will share best practices and standards for sample sharing and decision making. And we also hope to touch on the importance of discovering, integrating, and reusing physical samples for interdisciplinary work. At the same time, we would like to discuss the various challenges you have within your domain and share insights and resources in physical samples data management. We also prepared a collaborative document for today's session. This is accessible via the link, a link that my colleagues will share here in the chat. Perhaps Hazel can share in the link in the chat. Thanks, Hazel. The goal is to gather your experiences on how physical samples and data are managed, archived, and shared across your specific domains. So we invite you, feel free to add your input in that shared document. It's a crowdsourced document. Please add your name as well as your affiliation there so we can facilitate networking and collaboration among our attendees today. Let us know if you have any issues accessing the said document. All right, so that's our objective for today, for the meeting today. And now I have the privilege to introduce our first speaker. So our first speaker is Dr. Kerstin Lehnert. Dr. Lehnert is a distinguished leader in geoinformatics and scientific data management. She's recognized for pioneering open data and sample sharing in Earth and planetary sciences. She leads the NSF-funded Interdisciplinary Earth Data Alliance and NASA's Astromaterials Data System, contributing to over 40 projects within a number of agencies. Her extensive involvement includes serving on advisory boards for NSF, AGU, and NOAA on cyber infrastructure, data archiving, and access. And she has been honored in a number of prestigious awards. So today, Kerstin will talk about advancing fair and ethical data management for physical samples, the Internet of Samples, and the Sampling Nature Network. All right, Kerstin, please feel free to share your screen and go right ahead with your presentation.
You're on mute, Kerstin.
All right, sorry, that always happens when you. So I actually go back and just want to repeat that I'm really excited to be here because I think I'm probably going to learn more from you all than you from me. As far as I see, this is a really expert group coming together, and I hope I'm not just going to tell you what you already know. So my presentation I've basically structured into two pieces. One is sort of about fair samples and what can be done to make samples and sample data more accessible. In the second part, a very brief overview of the Internet of Samples and Sampling Nature Network, Research Coordination Network. So hopefully we will have enough time for questions later if there are things that I can't get to. I'm sure that everybody is aware who's working with physical samples and dealing with them in their data management activities or in their research, that science today demands access to samples and to the data generated from them. And it's, in part, it's that need for reusing samples, adding on to knowledge that already exists about a certain sample, about a certain location. It's also aligning the data generated from samples and any sample metadata that are available with modern-day digital and data-driven research and the opportunities that arise with new analytical capabilities. But the other thing always is the transparency and reproducibility of research. And you may be aware that some of the journals have actually started to demand access to the physical samples and not just to the data. Science is one of them. The other aspect is that really making samples and sample data more accessible and managing them in certain ways makes it possible to connect different data types that were generated on a single sample or different data types that were generated at a given location. And that's what I wanted to add to. And I saw NSF was here and said, yeah, there is, and somebody posted about the need for now sharing physical samples. I've just taken the NSF Earth Sciences sample and data sharing policy and NASA's policy in the planetary sciences because I'm most familiar with that as two examples, but I'm sure that in other agencies, this is very similar. So from looking at the interdisciplinary application of samples, the demand is clearly growing, but that requires us to be more consistent in the way we document samples, identify the samples, how we share them, and how we cite them also in the literature. And that is currently a huge problem because the landscape of samples and sample data is really highly fragmented, both with respect to disciplines that biology, earth sciences, archaeology have their own domain-specific best practices and standards and facilities. But then there is also a fragmentation across different types of institutions, agencies, and science programs, for example. But these principles, and now, I mean, this was already explained earlier, there are a number of principles that kind of help to converge all these different best practices. And FAIR was mentioned, CARE was mentioned. I'm also bringing up trust as, you know, a framework for the repositories to follow certain best practices. I'm not going to go any further into that. But when we apply FAIR to samples and sample data, there are a few real fundamental and requirements that can be applied across all domains. And that is that samples need to be documented with sufficient information so that they can be reused, but also that they can be found and made accessible. The second one is that they need to have persistent identifiers, globally unique, resolvable persistent identifiers. And this is exactly the same as with data. We have DOIs for data, and we have all kinds of metadata schemas to describe data in a proper way. The third is that, and this is through actually in many ways through these persistent identifiers and standardized metadata, that we can link the samples to the scientific interpretations in the literature and to the data that were generated from them. So again, just emphasizing the key to FAIR with samples really is the persistent and globally unique identification. Through the use of PIDs, we can ensure that samples can be tracked throughout their lifetime. And this is really something unique for samples. Samples live a long life. They go through many laboratories, they go through sample repositories and can be moved from a repository to a museum and so on. And in the past, the practice has unfortunately been to rename the samples very often, and the chain is broken in tracking the data. Through persistent identifiers, we can establish stable connections, and the emphasis here on stable, and to connect distributed data for a given sample, but also to connect data from a similar project or a specific cruise or field program with the samples. And most importantly, the use of PIDs or persistent identifiers enables attribution and credit for those who have worked on the samples, who have collected them, who have created the data, and who curate the samples as well. These are just two examples of what the current situation still is, and they just emphasize that the inconsistent naming or duplication of naming creates problems in the reuse of samples and in reproducibility of results. And I'm not going to read this. I can leave it to you to check the slides later, because I think it's going to be a long time otherwise. So with respect to the use of persistent identifiers, there are a number of options to use persistent identifiers for samples. I think that the most frequently used are the International Generic Sample Number and ARCs, which are archival, oh God, now I'm blanking out, archival key, I'm sorry that this happens to me. But anyway, so I wanted to show you how these identifiers can be obtained. So one place is the System for Earth and Extraterrestrial Sample Registration, but there are other places and other agencies that are using the IGSN to identify samples. The USGS, DOE is doing that, and in part, they actually using CSAR. Every registration generates a sample profile with as much metadata as is submitted by the user, the data curator, the sample curator, whoever it is, and they will obtain an IGSN that can then be used. And this is an example from a sample data system, a repository, where we are integrating these IGSNs, the persistent identifiers of the samples, into the metadata of a given data set, so that the connection between the data and the sample, as well as the actual journal paper, can be established in a machine-readable way. So I think this is a really important step that we have taken. So just to summarize, basically, the different steps that can help us get to fair samples and sample data. The first step is to really make sure you register your samples in a trustworthy, sustainable sample metadata repository and get a persistent identifier. Submit the data, manage them in a domain data repository, specifically using the persistent identifier to reference the sample data. And then ultimately, the samples also need to get submitted to a long-term sample repository. And that is actually one of the biggest challenges in the current landscape. We have very few sample repositories that are open to the entire community to deposit samples. Other challenges are that it's still a big step for researchers to follow these procedures, to know about them and to implement them in their daily research routine. The other challenge that we have recognized for quite a while is that there is too little collaboration and alignment of best practices across sample and sample data repositories. And that's where I wanted to briefly bring up these two projects that I've been involved with, the Internet of Samples and the Research Coordination Network Sampling Nature. Both aim to really advance the trends or interdisciplinary use of natural history material samples. And the iSamples project is basically building the technology, a cyber infrastructure that can be used across domains to support the management of samples or sample data. And the Research Coordination Network looks at the policy issues and tries to address the social issues of building networks across domains. The iSamples Internet for Samples, as I said, it's building a cyber infrastructure, and what it's basically what it's building is something that we call the iSamples in a box, a containerized system that translates metadata kept in local systems such as CSAR into a consistent structure, into a JSON LD schema with consistent metadata across for all the iSamples in a box that participate. Our initial set of participating organizations or repositories is CSAR, as I mentioned already, then the Smithsonian Institution, GEOM as a repository for metadata of biological samples and genetic sequences, and Open Context, which is a repository for archaeology and archaeological artifacts. We've put a lot of work into harmonizing metadata across these different domains, and there's simply not enough time to go into detail here. But we have identified that core set of metadata that can be applied across all the participating systems, and then ways to have extensions to these core metadata that are more discipline specific. For the Sampling Nature Research Coordination Network, I just wanted to emphasize its role in bringing together a very diverse interdisciplinary community of researchers that work with material sample data, where we're trying to really identify what are the barriers in all of these domains, be they technological, social, legal, and so on, to really equitably and justly sharing and reusing samples. And this goes a lot into the context of the CARE principles. We're trying to exchange and share information about the resources that exist across all these different domains and make sure that there is not so much duplication or development of new resources, new infrastructure where there is already something existing that can be reused. Ultimately, we're also trying to raise awareness, not only across the science community, but also in the general public about the value of material samples and their importance in societal, in research that addresses societal challenges. The Sampling Nature Research Coordination Network has had 3 workshops already that have been really successful in integrating these different communities, and a 4th one is coming up this May at the Smithsonian Institution. If you have any interest in this, please contact me and I will communicate back to you. The current focus I wanted to emphasize of the Research Coordination Network is, on the one hand, the implementation of the CARE principles, and we're working here with existing initiatives such as local contexts and the traditional knowledge labels, biocultural labels, to integrate the requirements and achievements from these efforts into the metadata of the Internet of Samples and the recommended sample metadata profiles. The other effort is that we really are concerned about the many, many uncurated collections in institutions all over the globe, where many samples that are irreplaceable and of high value are getting lost. So we're putting effort into initiatives that make these collections more visible, integrate them through registration in catalogs into a digital infrastructure where they can be more reused, can become more valuable. So where will all our samples go? That's basically the biggest question, I think. And I've just listed a number of resources here that may be of use for people afterwards. Thank you.
Awesome. Thank you so much, Kerstin, for the great great presentation. We accept your challenge to see whether you learn more from interacting with us or whether we learn more from you. I'm going to turn to a slightly different perspective. This is a sort of real-time laboratory that's actually dealing with the collection, the field collection of physical samples and the processing and sharing of those samples. So I'm thrilled to briefly introduce Lee Cooper and Alynne Bayard from the University of Maryland Center for Environmental Science, UMCES. Lee is a co-investigator on the Distributed Biological Observatory, among other projects. I see that Jackie's also on the line. Hi, Jackie. he has been an ocean-going researcher for a number of decades at this point, who focuses on how Arctic marine ecosystems and biogeochemical cycles are responding to environmental changes at high latitudes. Alynne Bayard is a faculty research assistant and the Swiss Army knife for the lab at UMCES. There, her work with Jackie and Lee focuses on mapping the team's data using geographical information system technology, as well as producing graphics, among many other hats that she wears. She also focuses on quality assurance for data that they share with the scientific community, the Pacific Arctic Group, among many other collaborators, which provides a point of connection to the specific topics we're going to cover today. With that, I will turn the floor over to Lee and Alynne to provide their remarks, and then we'll catalog your questions and think about how you want to tie these two presentations together, and we'll have plenty of time.
Great. Thank you, David. I'll go ahead and share my screen. I think we'll be able to give a pretty nice bookend to what Kerstin talked about, particularly the uncurated collection of samples, which is what we didn't start out with, this idea that we would be curators and provide samples, but it's actually become pretty important or a significant part of our job as to what we do as part of the Distributed Biological Observatory, which is an observational system. It's internationally coordinated through the Pacific Arctic Group. So all of the countries that are participating that send their ships through Bering Strait, we've got some agreements in place to stop and sample particular things, particular biological systems, so that we can track the how things are changing in this very important area. I might add, we're down in Chesapeake Bay. I've got a little arrow pointing to where our lab is located. So that's the mighty Patuxent River, which is the largest river within Maryland, and where it empties into Chesapeake Bay. And then beyond that, you could see the Potomac River and Virginia. So I'll go ahead and show you my own pictures of our storage building. These are sediment samples. You can see in the picture to the, I just went in there with my iPad and took a few pictures so you can get a sense of what we're dealing with. These are sediment samples that have been dried, and we've done grain size, or we've looked at the carbon isotopes or carbon nitrogen ratios. You can see the one box in the left, they're all labeled, so Jackie and I have a pretty good idea when we need to go find something where it is. That one says HX177, so that's an alpha helix cruise in 1993. So we brought all this stuff here to Maryland when we were hired here in 2008. So I hear about the storage building occasionally. So that's we also have a bucket room. We collect biological samples that are preserved in formalin initially. Then they're identified to species and then restored in isopropyl alcohol. We weigh them. This is how we're keeping track of the benthic animals in the northern Bering and the Chukchi Seas. What's the biomass per square meter, and are things changing? Is the community changing? And this is all as sea ice is retreating. And so these samples, once we're done with them, we're saving them. I'll get to it in a minute as to some examples of how we use samples or provide them to others for use in their own sampling. Here's an example. For example, this is an email sent to Jackie and myself in 2019 saying, hey, we don't have too many samples. These are people in Europe who were assembling a database for Arctic shelf sediments, and they didn't have too many examples of sediments from our half of the Arctic, and wanted to run some carbon-14 information on it. We also had carbon-13 data and organic carbon data. So what happened was, well, a couple of years went by. We sent the samples to Stockholm. They did their analyses. A paper was published, as you can see, and there's value added for us. Our names are on this manuscript, and I think that's been a pretty common pattern. One thing that happens, though, is now everybody references this paper rather than our original papers that describe some of the data. And I found that in some other cases too, but that's a minor thing, and it also shows that once somebody organizes an archive and publishes information on how to get that data, that more people tend to use it in the future. Here's another example. A graduate student at University of Chicago knew about the samples that we collect that end up in those buckets. So one of the things we do is if they're shells, or we're interested in the animals that are alive when we collect them, but they're little bits of clam shells and other things. And this student was interested, has finished now a PhD. And again, Jackie's name at least got on this paper, looking at the shells that were dead, and can you compare the collection of dead shells, the things that didn't have animals in it, with what we were finding in terms of the live assemblages, and what's the comparison? And could you go back, say, 100 years and say, well, this is probably what this community looked like at that time? And so this is a second example, and we think that all of the stuff that we do in terms of providing data also is pretty intrinsically linked to the samples that we collect, because when people get our samples, we still have to help interpret that. So I was going to turn the floor over to Alynne to talk a little bit about the challenges of how we provide data to others for them to use.
Yeah, so of all my Swiss Army knifeness, the dealing with the physical samples is not one of the tools. So I just wanted to give you a brief summary of the places that we have to submit our data. And for we have two main entities, so I put a couple of screenshots up there to illustrate that we have to submit our NSF funded data to the Arctic Data Center. And I suppose our NOAA data is meant to be at this the National Centers for Environmental Information. I'm more familiar with the Arctic Data Center, so I just ran through a little exercise to look up some of our data on the NCEI database. And it seems like they have several options, but it seemed like the Geo portal was the easiest one for me to find our data. Now, I have heard that people do have trouble finding data that they're looking for, or if they don't even know exactly what they're looking for, kind of browsing to figure out. So one thing that, we can go to
the next slide. Okay.
Yeah, so on the next slide, the Arctic Data Center, for example, has helped us develop, or helped other people to develop project-specific web portals. So we have one for the Distributed Biological Observatory, and the way that we have attached this to make sure, aside from the Arctic Data Center helping us tie these data sets to this portal, the main thing that we've been doing is making sure that all the people that are involved in this project work it's just as simple as making sure that you use keywords like DBO, not a word, but DBO, and Distributed Biological Observatory. And then that helps tie all of our data sets together. And I think we put an example of another international collaborator that.
Right, the Koreans have their own parallel system. So in the lower left-hand corner is their web page. And so part of what we do is because it's a distributed observation system, we depend on the Koreans putting their stuff in one place, the Japanese in another. We have our own requirements. And so the ultimate challenge really is to tie those all together so that you can find all of the data you want, regardless of who collected it. But this allows, and actually the Korean page is quite good in terms of the quality or how fast you can find things. So that's just another example. I'll just finish up with, so we can have some discussion with another challenge. For example, we do a lot of these projects at sea. This is on the Sekuliak last year, with infrastructure such as a sediment trap that's being recovered here, that's been purchased and deployed, and they're owned or the research is enabled by different entities. So for example, we have one of these sediment traps that has a inventory on it for the University of Maryland Center for Environmental Science. So they come around once a year and ask me, where is it? And I go, it's in the ocean. We're having this challenge in that there's different devices. We pull one out, put another one in, and we need to sort of develop a center to distribute samples for appropriate sampling. So this is actually the very first. It's not reusing samples, it's figuring out how do we distribute them. So in this case, Kerstin Lehnert, who's now working at the Korean Polar Research Institute, started us down this road of deploying sediment traps, and we've put them in a number of different locations now in our DBO network. But there are a bunch of people at a bunch of different universities that want little pieces of that, so they can do particular analyses, whether it's ice, sea ice, algae, biomarkers, or stable isotopes, or chlorophyll, or so this is a need we've got that's ongoing in terms of our current collaborations. Another collaboration is, of course, we go and visit some of our international collaborators. We do have meetings regularly of the Pacific Arctic Group, and I'll show, I think I've got a picture of one coming up. But this is an example. We went to Korea last May and went to Hanyang University, gave a little talk there about collaboration activities and how to help students if they wanted to come to the United States, for example, to identify advisors that had funding to support them. And while I was there, met a student who is doing some work that's pretty specialized, looking at compound-specific isotope measurements. And we do have a capability to do that here, but I don't have enough people to do that regularly. And by comparison, you can see from the picture, they have quite a big research group there in Korea, as opposed to what we have here in the United States. And so I said, but their other problem is they send the Korean ship out and they don't get as many samples as we do. So I have a lot of samples. They've been identified at sea. They've been freeze dried. They're ready to do analyses. And I said, well, we could probably give you some. And so we're working on that. Actually, I'll see this student at the Arctic Science Summit Week in Edinburgh next month, and we're going to work out, what exactly would you like, and we'll say I'll ship them to you. So that's something that's ongoing right now. Jackie gave me this one slide that I thought I would add, and these are just some publications that are over and above what we do as part of our work anyway, that have come out of this process of being willing to share and give samples to people when requested and make them openly available. And you can see most of the time our names get attached to the papers that come out of this, so it's win-win for us. So in just a brief conclusion, here's our Pacific Arctic Group meeting in 2022. There was a meeting in the fall of 2023 also in Tokyo. So we moved this around, both hybrid and in person. I think everybody agrees that sharing the samples, it adds value, and we use the Pacific Arctic Group and other international frameworks. They're good places to identify these opportunities for sample and information sharing, but there's little funding and a little forethought that's gone into this. That comes back to our sample collection in the storage building here, and we also recognize we need to move to better coordination with all of our funding sources and the expensive collection platforms, whether they're water samplers or sediment traps or going out to sea on expensive ships. And my observation is the archiving data is also neglected, and even if it's archived, we must often explain the data to users and budget enough time to help. And I've just been working on a paper I'll give a talk on next week, and finding problems with data that was collected, little spots, or this this doesn't make sense. So we'd never really budget enough time for quality assurance of our data, as well as taking care of the samples. So I'll stop screen sharing so we can, let's see, close, stop share, there we go. And thanks for your attention.
Awesome. Thank you so much for the amazing presentations. And clearly, I'm a little bit overwhelmed to think about all the different topics that both of the presentations have teed up. But I'd love to hear what questions folks have, at least as an initial response. And that can be anyone that's on the that's participating in this meeting. So please feel free to raise your hand or offer comments or questions in the chat, and we will run from there.
Yeah, and we also encourage our speakers to ask questions between the two of you, the two groups, so we can maybe that can inspire discussion points as well. Yeah.
And then I just start if nobody says anything. It was such a typical picture there that Lee showed of, the uncurated collections. I've seen a lot of those. Is there actually a digital catalog of everything that is in that facility?
No, and part of that is just, the capitalization, I think, that would have to go into it. I mean, we have boxes organized by crews, say, and so I have a pretty good idea. I could find something in 10 or 15 minutes. But to go back to that example from the Stockholm, they said, oh, we'd really like these samples from the Russian Arctic. We'd been in a couple of times on the Russian side, and I had a hell of a time finding those. That some things that we thought we had, something happened to them. I saw your identity or the paperwork that you might write up for geological samples or sediment samples, and I just I looked at it and I said, we're not doing that.
That's why I'm asking specifically, because through these, efforts in the Sampling Nature Research Coordination Network, and where we work with this tree ring lab at Lamont here in my home turf, so to say, we realized that, the effort is simply too big to catalog every individual sample. So what we're going to experiment with is a registration of collections in the first place, right? That you at least describe a set of samples that was collected at a given research cruise or at a field program, or that has been collected from different researchers together as some reference collection and so on, so that at least the record is there and accessible, that these samples exist, that there is something. And then, as these collections maybe start to be used, then you can start also registering the individual objects as they get analyzed, because probably not every sample that's sitting there will ever be used. But those 3 samples for which isotope analyses are done, those 3 can be registered and then properly referenced in the papers. So that's sort of a practice that we're starting to investigate. And I would love for anybody to be a guinea pig here and come and join us and, kind of work with us on testing that out, if that is something that can move us forward.
Go ahead, Sal.
Yeah, it'd be great to move in that direction. Yeah.
We could do the question in the chat first, Cindy, and then I can ask mine.
All right, so Tim, You can also unmute if you would like to ask your question.
Sure. So I was wondering what functionality do any of these various sample registration sites offer with respect to subsampling? You know, I can see a given big sample being taken apart and used for different purposes, you know, DNA over here, something else over here. How do you maintain the linkages between these various subsamples? Or are they all assigned the same IGSN?
Oh, sorry, I was distracted in answering a question in the chat.
That might have been my question.
Are you with linking samples?
Uh-huh.
Yeah.
Yeah.
So yeah, I mean, I was just typing. You can link them at any level, you know, in multiple layers. So we have a lot of registrations from the ocean drilling program, and they registered sort of the top level is the whole. Then the cores, core sections, core halves, you know, it goes down to some, so you can have 6 layers. And because the tree is really tracked, you can see the siblings, you can see, you know, so that's all really taken care of. It's just a practice that you have to follow to start registering the objects from the top, right? So you can enter the parent of a given sample into the metadata of the child.
Well, thank you. That all sounds very encouraging.
Yeah, so my question was, I think following up on Lee's picture and sort of a lot of people, I think, thinking about the overwhelming nature of trying to catalog all their samples or do this for all the samples they have. Do you have any other suggestions on how people can start? Like, what are some first steps, or should, you know, ways to break it down just to make it feel a little more manageable for people?
Yeah, I mean, as I mentioned, one thing is to really start with cataloging those samples that are taken out of the collection for doing analytical work. That's, I think, the most important step. Everything else that just sits there can maybe at some point slowly be taken care of. But to me, you know, the access to metadata of the sample and access to the facility where the sample is most important for those samples that get published in the literature. So you can go back for reproducibility reasons, but also to say, oh, you know, now there's a sample that, you know, helium isotopes were measured on, so now I want to analyze neon isotopes in those samples as well, right? So it actually builds much more comprehensive scientific data sets.
Yeah, so I think the idea that people can start small and they don't have to do everything at once is a nice one to think about. Thank you.
Yeah, go ahead, David.
Actually, I feel like I have more access than others, so I'd love to turn to Leandra's question first, and if we have time, then I'll come back to mine.
All right, sounds good. Leandra?
Hello, good morning and good afternoon for some of you. This is Leandra with the North Slope Borough. I have a question for the second part of the presentation in regards to authorship of manuscripts and how that works with using the samples, because usually that's very a point of contention on things like losing the thread from when the samples were collected until someone else uses it. So when there is a direct connection, for example, you gave Lee that you and Jackie are part of the same project for many, many years, and there is a continuity, you kind of can keep a pulse on how those samples are used and analyzed because you are the main point of contact. But for many projects and institutions, that's not the case. And credit or lack of credit might end up being lost in the way in the process when these samples are collected and used for other purposes. So just a discussion for the group or something to consider. What are the challenges and solutions that have been found so far? Thank you.
Well, I could just briefly respond. I mean, whenever we contribute intellectually to something, then we feel like, well, we have some stake in the project. So it could be an acknowledgement. It might be sufficient, but you're right. I think a lot of times if people don't understand entirely where did this sample come from, or who collected it, who was out on the back deck, put it in a bag, identified it, made sure it was handled properly. Because even now we transfer samples, say, for example, to the harmful algal blooms, people who have the interest in that. And I don't think it's always commonly known, everybody who's involved in the chain of custody, to getting that sample to where it needs to go to get analyzed. So it is a challenge that we all have all the way through the whole process.
Thank
Maybe
you.
I can just add, I know that some journals have actually established these roles in an authorship, and the contributor of sample is part of that. And there has been, I'm not sure who is familiar with the Research Data Alliance, there was a working group quite a number of years ago, like 5-6 years ago, a working group on providing credit to sample curators and sample collectors. So it's worth looking. There was a publication coming out of it, but I just don't remember the exact where. I think it was Anne Thesson. But people are thinking about it. And I think that's where it's also so important to have the metadata. Who was the sample collector? Who is the contact now in the metadata of a sample registration, accessible to everybody, so you can track that.
Thank you.
Hazel, do you want to jump in?
Sure, I'd be happy to. I was actually just going to ask, I've just looked around at, again, through the introductions that folks gave at the beginning of this call, and this conversation is really powerful, and there's obviously a lot of expertise in the room. So I'm wondering, the data and MOP teams had prepared this collaborative document for folks to work on here that will help. I think Cindy and David had talked about how this might be a meeting series that sort of builds over time. And I wonder if we want to spend a minute there. David, I don't want to cut in on your question, but if we want to make sure to put aside a minute or two to get folks working in that document so we can start to build momentum towards future series as well. Cindy or David, what are you thoughts on that?
Yeah, absolutely. So the goal for that document is really acknowledging that here we have participants coming from different domains. We heard from the marine domain through Lee and Alynne, but acknowledging that there are others here who are in, other fields that perhaps, no, I'm sure, have their own best practices, different protocols and different tools. So this is just, hopefully our goal here is to be more of resource sharing, as well as for networking and collaboration, so we can move forward with this topic as a group, as a collaboration team, right? So yeah, so that's the goal there. I'm gonna stop talking now, and if you have a minute to share or copy paste any resources, best practices you have in this document, feel free to do so. And again, reminder to add your name and your affiliation or even your email address so we can reach out to you. So just, yeah, thanks. David, do you want to share other thoughts?
No, I think it's always great. And Hazel, thank you always for such really thoughtful and timely interventions. Folks, please add your ideas. We want this to be a community-driven activity and informed by and therefore of interest to you. So please add your ideas to the to this crowdsourced documents, and thank you for bringing our attention back to it, Cindy and Hazel. I just wanted to ask a quick question as well, since I have the floor, and that was really to I think we've already asked you this because under Cindy's tenure as a Knaus Fellow, we did a lot of work exploring data practices with all of our PIs within the Arctic Research Program. But I just wanted for the group, you mentioned budgeting for data collection and data management, and it's clearly a growing issue. For the group, can you give a sense of what you typically budget for data management and data collection, and an idea of what that number maybe should look like as a percentage, given the current expectations?
Oh, I don't think we ever budget enough. But you know, if you look at your time you spend looking at data sets and trying to rubbing your eyes and trying to figure out, do I believe that number, or, you know, going back and then finding Excel spreadsheets that don't have columns aligned right or something like that. I suppose we probably, you know, and I include technical assistants like Alynne here, that we probably budget 10 to 20% of our time for looking at the data sets after they're generated. And maybe that's not enough.
Yeah, and since the physical samples topic is definitely something that we're all still thinking about, and it's a big challenge, we would also appreciate, I guess, in the program management perspective, thoughts on how we can improve the data management planning system. What are the missing pieces in the data management plan that, you know, the templates that we provide to the PIs? So yeah, so maybe perhaps we can think about those too, so we can improve our processes. Yeah. Any other questions from the group? I know we're at time, but maybe last question from our audience, from our participants. All right, that was 6 seconds, so that's great. Hazel, any other thoughts? Maybe we can hand it to you to close the meeting.
Nothing big. I just put a note in the chat sharing where the recording will be posted. The collaborative document link is also posted at that site, so please do feel free to keep editing that. This was a really excellent meeting, so just a big round of applause for the speakers and for the team leads from the data and MOMP teams for hosting this really interesting discussion. I hope you all have a wonderful Wednesday.
Thank you.
Thanks so
This
much,
was
everyone.
great.
And look for additional announcements coming up, because I think we're clearly just touching the tip of the iceberg, to use a I don't know. Anyway, please look out for additional announcements, because I think this is a really important topic and we have a lot of ideas to explore. Thanks.
Thank you.
Thank you.